It's me Joseph, your new chemistry teacher. We are starting with our new chapter, biomolecules. This biomolecules comes under the branch of science which is biochemistry. Biochemistry means the chemical reactions and the chemical compounds that involved in the biological functioning of a living organism. So the study of chemical processes that involved in the biological functioning of a living system is called biochemistry. And we know that human body or any other living system is composed of a large number of complex molecules, and such complex molecules are called biomolecules, the molecules which have some biological functions. So such molecules are called biomolecules, and the main examples of such biomolecules are carbohydrate, proteins, amino acids, lipids, vitamins, etc. So in this chapter we are going to study the structure, properties, the functions, and some of the chemical reactions of certain biomolecules like carbohydrate, protein, and amino acids. The biomolecule is carbohydrates. The major examples of carbohydrates are glucose, sucrose, fructose, cellulose, starch, etc. So we have to understand what is this carbohydrate, from where we can obtain this carbohydrate, and why it is considered as a biomolecule, or what is the biological function of this carbohydrate. First, what is carbohydrate? In earlier times, these carbohydrates were defined as the hydrates of carbon with the general formula CxH2OY. This is hydrates of carbon. So all the compounds that corresponds to this formula were identified as carbohydrates. For example, glucose. The formula of glucose is C6H12O6. That corresponds to this formula C6H2O6. Similarly, sucrose with chemical formula C12H22O11. That corresponds to this formula C12H2O11. So such compounds were considered as carbohydrates. But later it was found that some of the carbohydrates cannot be fit into this formula. For example, rhamnose, whose chemical formula is C6H12O5. And this carbohydrate do not correspond to this formula, but it is a carbohydrate. Moreover, some of the compounds which are not carbohydrate corresponds to this formula. For example, acetic acid, that is CH3COH, which is acetic acid. Its chemical formula will be C2H4O2, that is C2H2O twice. So this acetic acid corresponds to this carbohydrate formula, but it is not a carbohydrate. So nowadays, carbohydrates may be defined as polyhydroxy aldehydes or ketones, or compounds that can produce such units on hydrolysis. Now we have the answer of our first question. What is carbohydrates? Carbohydrates are polyhydroxy aldehydes or ketones, or compounds that can produce such units on hydrolysis. So our next question is, why it is considered as a biomolecule? It is because it has some biological functions. The main biological functions of carbohydrates are, it acts as a source of energy as well as a storage of energy. Source and storage of energy. Sugars that comes under the category of carbohydrates are an excellent examples of source of energy. Examples are sucrose, lactose, etc. They are sugar molecules which are also carbohydrates and act as a source of energy. Again, these carbohydrates can act as a storage molecules of energy like starch and glycogen. Starch is the storage molecule of energy in plants, and glycogen is the energy storing molecules in animals. So this is the main biological action of carbohydrates. It can act as a source as well as storage of energy. Our next topic is the classification of carbohydrates. Carbohydrates are broadly classified into three groups depending upon their behavior on hydrolysis. They are monosaccharides, oligosaccharides and polysaccharides. Actually, saccharides are the another name of carbohydrates. It comes from Greek word saccharon, which means sugar. Most of the carbohydrates are sweet in taste, so they are also called saccharides, meaning sugar. So these carbohydrates can be classified into monosaccharide, oligosaccharides and polysaccharides based on their behavior on hydrolysis. First one is monosaccharides. Monosaccharides means the carbohydrates which cannot be hydrolyzed into smaller unit. Such carbohydrates are called monosaccharides. Example, glucose and fructose. Next, oligosaccharides. Oligosaccharides means the carbohydrate which can be hydrolyzed to form 2 to 10 monosaccharide units. Such carbohydrates are called oligosaccharides. Sucrose, maltose, raffinose, etc. are comes under oligosaccharide group. Sucrose, its formula is C12H22O11 and which on hydrolysis gives glucose and fructose. Both have the same formula but their structure is different. So this sucrose on hydrolysis gives 2 monosaccharide units. So it comes under oligosaccharide category. Again, maltose. Its chemical formula is C12H22O11. This chemical formula of sucrose and maltose are same, but their structures are different. They are entirely different compounds. And which on hydrolysis gives 2 molecules of glucose. That is 2 molecules of monosaccharide units. So these two these two carbohydrates are oligosaccharides, and again it can be termed as disaccharides because it gives 2 molecules of monosaccharides on hydrolysis. Again, another raffinose. Raffinose, its chemical formula is C18H32O16, which on hydrolysis gives galactose, glucose and fructose. 3 molecules of monosaccharides. So this raffinose gives 3 molecules of monosaccharides on hydrolysis. So it comes under the category of oligosaccharide. Again, it can be termed as a trisaccharide because it gives 3 monosaccharide units on hydrolysis. So any carbohydrate which can give 2 to 10 monosaccharide units on hydrolysis are called oligosaccharide. Again, last classification is polysaccharides. Polysaccharide means the carbohydrates which on hydrolysis gives large number of monosaccharide units, more than 10, large number of monosaccharide units. The examples are starch and cellulose. And these polysaccharides are also known as non-sugars because most of the carbohydrates are sweet in taste, but these polysaccharides are not sweet in taste. So they are called non-sugars. Carbohydrates are again classified into 2 types, reducing sugar and non-reducing sugar on the basis of their action on filling solution or tolerance reagent. This is a reducing agent and only aldehydes or ketones group can reduce this filling solution or tolerance reagent. And we know that carbohydrates are polyhydroxy aldehydes or ketones. So they have aldehyde or ketones group in it. So it can easily reduce this filling solution or tolerance reagent. So the carbohydrates which can reduce filling solution or tolerance reagent are called reducing sugars, and the carbohydrates which cannot reduce filling solution or tolerance reagent are called non-reducing sugar. All monosaccharides are reducing sugar like glucose, fructose, etc. Because in the case of monosaccharide they have free aldehydes or ketones group and that can reduce this filling solution and tolerance reagent. So they act as reducing sugar. In the case of disaccharides, they are formed by combining 2 monosaccharide units. On the combination of that monosaccharide units, the aldehydic or ketonic group may get bonded. That creates an unavailability of free aldehyde or ketones group. So such carbohydrates or such disaccharides cannot reduce filling solution and tolerance reagent because they have no free aldehyde or ketone group. So sucrose is an excellent example for a non-reducing sugar. But other disaccharides like maltose, lactose, etc. are reducing sugar. but this sucrose is non-reducing in nature. Let's discuss monosaccharides in detail. First, its classification. These monosaccharides can be broadly classified into two categories. First, on the basis of the functional group present in it, whether it is aldehyde or ketone. Second, on the basis of the number of carbon atom. If the monosaccharide units contain aldehyde, CHO group as the functional group, it is called aldose. Or it contains ketone, it is called ketose. So this is the first classification based on the nature of functional group present in it. And second, on the basis of number of carbon atom. If the monosaccharide unit contains 3 carbon, it is called triose. 4 carbon, it is called tetrose. 5 carbon, it is called pentose. And 6 carbon, it is called hexose. Again 7, it is heptose, and so on. This is glucose, and it contains 6 carbon atom. So glucose comes under the category hexose. And the functional group present in glucose is aldehyde, so it is also an aldose. Again, fructose. The formula of fructose is the same, and this is fructose. The only difference is it contains the functional group ketone. So fructose is a ketose and glucose is an aldose, and both are comes under the category hexose because the number of carbon atom is same in both glucose and fructose. In this chapter, we have to study the preparation methods and structure of two main monosaccharides, glucose and fructose. First, let's consider glucose. These are the two methods of preparation of glucose. Glucose can be prepared from sucrose as well as from starch. First, from sucrose. Sucrose means sugar, cane sugar, and its chemical formula is C12H22O11. And this disaccharide or this cane sugar on hydrolysis in an acidic medium gives two monosaccharide units in which one is glucose. So this cane sugar on hydrolysis in an acidic medium, we can use either hydrochloric acid or sulfuric acid, in an acidic medium gives two monosaccharide, glucose and fructose. So this is the simplest and the first method of preparation of glucose. Second, glucose can be prepared from starch. Starch is a polysaccharide unit, and this N number indicates a large number of monosaccharide present in this polysaccharide starch. And this starch on hydrolysis in presence of an acidic medium at 393 Kelvin temperature and 2 to 3 atm pressure, it gives N number of glucose units. Okay, so starch is a polysaccharide which contains large number of glucose units in it, which on hydrolysis in an acidic medium at 393 Kelvin temperature and 2 to 3 atm pressure, it gives a large number of glucose units. So these are the two methods of preparation of glucose. First, from sucrose, it's a disaccharide. Second, from starch, it is a polysaccharide. Both on hydrolysis in an acidic medium gives glucose. Our next topic is the structure of glucose. The structure of glucose was elucidated on the basis of certain evidences. That means by analyzing the product formed on certain reactions of glucose. Before that, we know the molecular formula of glucose. It's C6H12O6. And glucose is a carbohydrate, and carbohydrates are polyhydroxy aldehydes or ketones. That means it contains 6 carbon, and there is an aldehydic or ketonic group in this glucose, and there are also OH group because polyhydroxy aldehyde or ketones are carbohydrate, and this glucose is a carbohydrate. So this is the information we obtained from its molecular formula. It contains 6 carbon. There is an aldehydic or ketonic group, and there are some OH groups. Next, scientists has analyzed the structure of product formed from glucose on treatment with hydrogen iodide. Hydrogen iodide is a reducing agent. It can reduce aldehyde, ketones, alcohols into hydrocarbons. So this is an expected structure of glucose. We know that glucose is an aldose, so there is an aldehyde group, 6 carbon and some hydroxyl group. So it's a polyhydroxy aldehyde, it's glucose. And when it is heated with hydrogen iodide, it forms normal hexane, that is 6 carbon in a straight chain. So this is a hydrocarbon normal hexane. From this product, we can understand that the 6 carbon present in glucose are in a straight chain. So this is the truth indicated by this product, 6 carbon in a chain. From its molecular formula, we get there are 6 carbon. From this reaction, we get another point, that is 6 carbon atom in glucose are in a straight chain. Next, the scientist wanted to know whether there is a carbonyl group in glucose. For that, they carried out certain reaction which gives result only for aldehyde or ketones. These are the two reactions. First one, the reaction of hydroxylamine with glucose. Hydroxylamine means replacing one of the hydrogen by an OH group in ammonia. Then we get hydroxylamine NH2OH. And this hydroxylamine reagent reacts with carbonyl group, forms oxime. Here, the oxygen from the carbonyl group and H2 from hydroxylamine get removed as water, and the remaining portion CH double bond NOH is the oxime functional group. So the formation of this oxime product indicate the presence of carbonyl group in glucose, because the oxime is formed only when hydroxylamine react with a carbonyl group. So this reaction indicate, or this reaction proves the presence of carbonyl group in glucose. Again, when glucose is treated with hydrogen cyanide, it forms cyanohydrate. And this reaction is also an indication of the presence of carbonyl group. Here, we know that these are the charges of ions, hydrogen and cyanide ion. And here, oxygen is more electronegative, so this pi electron shift towards this oxygen. As a result, this oxygen get a partial negative charge and this carbon with a positive charge. So the hydrogen comes to this oxygen and form OH, and this negative cyanide group comes to this carbon. So this is the product, cyanohydrine, CHCnOH, CHCnOH. So the formation of cyanohydrine indicate the presence of carbonyl group in glucose. So these are the two reactions which give a firm evidence for the presence of carbonyl group in glucose. So we get the points. First one, there are 6 carbon atoms. Second, the 6 carbon atom in a straight chain. Third, there is a carbonyl group in glucose. Next, we have to understand the nature of carbonyl group present in glucose, whether it is aldehyde or ketone. Both contain carbonyl groups, so both show almost similar pattern of reaction with most of the reagents. But there is a difference in their action when we use mild oxidizing agent. Aldehyde undergo oxidation reaction in presence of mild oxidizing agent, but ketone did not give any products under the influence of mild oxidizing agent. So aldehyde is reactive towards mild oxidizing agent and ketone is unreactive. So we can identify the presence of aldehyde or ketone, or we can distinguish the presence of aldehyde or ketone by using a mild oxidizing agent as the reagent. So here glucose is treated with bromine water, which is a mild oxidizing agent, and we get the oxidized product gluconic acid. This aldehyde group is oxidized to carboxylic acid group, and the product obtained is gluconic acid. Here the oxidation is occurred, means the carbonyl group present in glucose is an aldehyde, because only aldehyde can undergo oxidation reaction in presence of mild oxidizing agent. So this reaction indicates the presence of aldehyde group in glucose. Next, we have to identify the number of hydroxyl group or the alcohol group present in glucose. We know that glucose is a carbohydrate, so it is a polyhydroxyaldehyde, and we have to identify the actual number of hydroxyl group present in glucose. For that, we are using acetylation reaction. Acetylation reaction means an alcohol is treated with acetic anhydride, and we get an acetate compound and carboxylic acid group. And here, this is a ethanol, an alcohol, and this bond is polar. So this oxygen is more electronegative, so it will get a negative charge and it get a positive charge on breaking that bond. And here also this carbon oxygen bond get broken, and this carbon get a positive charge. Since it is less electronegative than oxygen, it get a negative charge. So this negatively charged oxygen combines with this proton and form carboxylic acid, and this CH3CH2O group combines with this COCH3 group, and we get the acetate derivative. So this is the simple acetylation reaction. When glucose undergoes such acetylation reaction with acetic anhydride, we get a pentaacetate derivative. So we get penta-acetate group. That means there are 5 OH group in glucose. So the formation of this penta-acetate product on reaction of glucose with acetic anhydride indicate the presence of 5 OH group in glucose. So we get another factor about glucose, that is there are 5 OH group in glucose. And here this 5 OH group remains in different carbon atom. That is why it is stable. If there is 2 hydroxyl group in one carbon, it will get easily hydrolyzed and form some carbonyl compounds. So the penta-acetate derivative product indicate the presence of 5 stable OH group. So in order to make it stable, this 5 OH group should be present at 5 different carbon atoms. Next is the reaction of glucose with nitric acid. Nitric acid is a strong oxidizing agent. It can oxidize both carbonyl groups and alcoholic groups into carboxylic acid. So when glucose on treatment with nitric acid, it gets oxidized into saccharic acid. It's a dicarboxylic acid. So the oxidation product of glucose is saccharic acid. And this product is obtained also when gluconic acid is treated with nitric acid. So both glucose and gluconic acid yields the same product, saccharic acid. Both gives same product means the last group present in glucose and gluconic acid are the same. Therefore, there is a primary alcohol in glucose as that of in gluconic acid. That is why it gives similar product on oxidation. So this reaction indicates the presence of primary alcohol in glucose. Okay. From these evidences, we can elucidate the structure of glucose. The molecular formula of glucose is C6H12O6. There are 6 carbon atoms and the 6 carbon atom in a straight chain. So there are 6 carbon atoms in a straight chain. And there is a CHO group, aldehyde group in glucose. So there is an aldehyde group. There is a primary alcohol in glucose. So there is a primary alcohol. And there are 5 OH group, 5 hydroxyl group on each carbon atom. So there are 5 hydroxyl group. The remaining valency of carbon can be satisfied by placing hydrogen. So this is the structure of glucose, C6H12O6 with 1 CHO group, 1 primary alcohol group and total 1, 2, 3, 4, 5 OH group. We can see that the position of hydroxyl group is not the same. Some hydroxyl groups points towards the right and some others points towards the left. So there is a difference in the position of hydroxyl group on the carbon chain. Why it is so? So tomorrow on the next class we will discuss why there is difference in the position of hydroxyl group in glucose and how can we identify the exact position of hydroxyl group in a glucose chain.